So photosynthesis, where it said these are, there was a question about reactants and products. Here are the reactants of photosynthesis. Here are the products of photosynthesis. Where it occurs in cells, and then we got to like the organs. So if we're talking about plants, we said the leaves are the photosynthetic organ of the plant, and now we're going to draw a heart of a leaf. Okay, so if we take, oh, that's a hydrogen ion. Here you go. Yep. For photosynthesis.
Good second catch.
Yeah, third, fourth. Okay, so cross section of a leaf. So if we look at the top of a leaf, we take a leaf, and we cut it, and then look at it under the microscope. What do you notice about, without putting it under the microscope, what do you notice about, like, which side is always greener? The top. All right, so why do you think the top of a leaf is always greener? There's more chloroplast. So Faith, why would it be helpful to have more chloroplast on the top of the leaf versus the bottom? So there's a waxy surface on top to protect it from that. But why would the top part be greener? Because he said there's more chloroplast. And what are chloroplasts going to do? Say that again. It's going to capture more sunlight. So it doesn't do you any good if you have more chloroplast beneath it, because the sun doesn't shine underneath the leaf. So that's important when we actually draw this out. So this top layer, so here's the top of it. The bottom layer is going to be down here. So the top is, there's a waxy cuticle. We will come back to this when we look at plants. We'll re-loop it back, and then we'll talk about photosynthesis one more time. That way we can make sure that everything's solidified before you take an exam. But that is an adaptation. It's a characteristic these plants have inherited. Why would it be helpful? I know Faith said about, it gives a protective waxy layer. What else would putting wax on the top of the plant where you're going to have more intense sunlight help that plant out?
Not drying up. Right.
So it helps with water retention. So waxy cuticle on top. Beneath that layer, oops, what happened? Go back. I don't want that side. Uh-oh, how do I make it big?
No. We didn't know that. We had no idea.
How do I do it? And I just,
Is it right there?
How do I do it?
Arrow
This
right
What you had to do.
here. Oh, everything is moved to the bottom.
What I'm saying is, you didn't give her directions.
I don't know what happened.
No, you're doing it wrong.
Oh.
How you do it?
Okay. Here we go.
Whoa.
Possessed board. Okay. Well,
How's the day been, bro?
all right.
you know.
All right. So the top part is beneath the waxy cuticle is the epidermis.
Hasn't been fun.
Epi on top, dermis is skin. You have the same stuff. That's this top layer of your skin. So also on the bottom, there is a epidermal layer too. But they have these little moon or like little apostrophe or even parentheses looking cells. One is on each side of a hole. All right, so this is at the bottom. There's still some epidermis, but there's holes in the epidermis. Okay, so if the top part of the leaf is the greenest, if we look at it under the microscope, we have all of these cells. So these are all the same plant cells that we have looked at, and we would find all those same parts that we've identified. But they seem to have a whole bunch of chloroplast. So what I'm not drawing is mitochondria and a nucleus and ribosomes and rough yard. I'm not drawing any of that. All I'm going to do is just draw some little chloroplast in there. So we'll just kind of pack them in here. Now, your test is black and white. So if you see a lot of little dark ovals inside of a plant cell, they're telling you those are chloroplast. Okay, just know that's, and you'll see that on a multiple choice question that you have in your packet. But all of these, here's that greener part. Now the difference in this section and the next section is that these cells are all kind of scattered around.
That's the only thing you missed. That and then the bottom.
We're going to put one circle right here. So I'm kind of leaving a little space. So technically, these are green too, because the bottom of the leaf, some have adapted not to have that color on the bottom. Like there's a magnolia out there that's brown and fuzzy on the bottom. You don't see any green at all. But there is, and then other leaves are a light green on the bottom and a dark green on top. And it's just because these cells that are scattered through here have chloroplast, because some light diffuses and gets down, penetrates that top layer and can still make its way down, but there's less. So why in the world do you think, if this top layer is most photosynthesis, so this is mesophyll, it's also actually called the palisade parenchyma mesophyll. Y'all just have to know mesophyll for right now. So think meso means middle. It does not want me to write a Y. There we go. All right, mesophyll. So there's most photosynthesis is there. What was the question? Is there a question? No. Okay. This bottom also is mesophyll, but there's all these air spaces. So why in the world would there be air spaces inside of a leaf?
For oxygen to fill up.
Okay, so what gas is coming into the plant? Okay, and then we have oxygen being produced that needs to leave, right? So here's how this works. These holes right here. What do you say, Taj?
The gas stations.
Kind of. All right, that little hole is a stoma. So it's like your nostrils. So you have this hole and you've got these little side parts of your nose that close and open and close and open. Well, we've got these little guard cells that do the same thing for those holes. So if the holes are open, then that allows that gas exchange to occur. But if it's really hot outside, I don't want those holes open. I don't care if I have a lot of sunlight and it's in the heat of the day. I want to shut that down, because what also can I lose out of those holes?
Water.
Water, evapotranspiration. That was part of the water cycle. So I want to retain that water, but the problem is if I keep this closed, I can't uptake CO2. I can't let out all that oxygen. If oxygen builds up inside of that leaf, that is not a good thing. So, so what's interesting is some plants have actually adapted to that. So desert plants mostly don't act like this. They photosynthesize at night. What they do is they store up what they get during the day, and then they open up the holes and take in their CO2 at night so their photosynthesis can take place. They reverse the order. But for most plants, this is what happens. So the stoma are going to allow for gas exchange. So I'm just going to put holes for nostrils and gain CO2, release oxygen, but also water, unfortunately, can escape. So in here, we also have, we'll move that over. We also have a vein. And in that vein, just like for you, into your blood vessels, you have vascular tissue. Blood is a tissue. So what does your blood carry for you? Okay, so your red blood cells are going to be your gas movers, oxygen and carbon dioxide. All right, what else? What else does your blood carry? Nutrients.
Am I right or wrong?
Yeah.
Hemoglobin.
Well, so that's in your red blood cells. But yeah, we're talking about hormones and all those little tiny monomers. All these little itty bitty monomers that are now carried in your bloodstream so they can be uptaken by the cells or taken up. So then you've got waste out there, and most of your blood is water. So that's the same stuff that's inside here. Right there is a vein with vascular tissue. And that's the word xylem and phloem. So there was two of the choices, I think, on that little Quizlet or quiz is. So xylem and phloem. And they'll come back up later, but we know that these have been on our exam numerous times because they equate it with having a circulatory system like blood. So xylem carries water and phloem carries sugar, the glucose that's made. Okay, so that's the anatomy of where photosynthesis takes place. And if we understand that, then we also know, are the leaves always green? So there's some other colors, some other pigments that their job is to attract sunlight. So what happens to chlorophyll in the fall, at least around here? Starts to break down, and then you start seeing other colors shine through. Well, those colors are also in here, locked in those chloroplasts. They're in there, you just can't see them. So there's yellows, xanthophylls, there's carotenes, that's why carrots get their name. There's also anthocyanins, the reds and the blues. So their job is to trap sunlight too, as the sun and the earth starts to change position. So it gets colder in the fall and the winter, but also, you know, our rotation is we're farther away from the sun, right? So the light's not as intense. So what can we say about photosynthesis in the winter and the fall, at least around here? The rate goes way down, and in the wintertime, it goes way, like, the lowest because we don't have all these leaves that are on those trees. And some of the trees do. But anyway, so that's kind of how it works. We're going to go inside of one of those chloroplasts, though. So we're going to draw a chloroplast. That's the next thing we have to draw. And when we do, leave some space above it and below it and make it big enough to where you can write something on the inside. I'll take
conifers, trees that stay around.
There are some flowering plants too that retain their leaves, and in fact, there's several shrubs out there that still have leaves that are flowering, but conifers all do, evergreen. Yeah. Okay. So here is how it occurs. So how. Okay, so we are going to draw this chloroplast. So let's do it like this. Now, chloroplast is also made of a lipid bilayer, and it's got proteins embedded in it, just like the cell membrane. We're not going to draw all that. Y'all don't have to know that. So we've got these little discs inside of it. When you look at this chloroplast, technically it is not green on the outside. Even though when we draw it, it's going to be green. What makes it green are these. Yeah. So these little stacks of like green pancakes or green hamburgers, those are actually the thylakoids, and that's where the chlorophyll is. But all this space out here doesn't have any green. So we're going to take your green, got one of those, and we're going to go ahead and kind of draw these little stacks. Okay, and you can color them in or not, either one. These are the thylakoids. Okay, so the thylakoid membrane or the thylakoids. So we have a stack equals a granum. So if you see this word, grana or granum, it just means like a stack of those thylakoids. Each one's like one little packet of chlorophyll. So we'll put with chlorophyll. But like I said before, there's other pigments, accessory pigments that are there. So that's what we did in this lab over here is we actually separated on Friday chlorophyll. We crushed a bunch of chloroplasts and released the pigments, and they separated them. And all they could see was a bunch of green. They saw two colors of green. They saw a yellow and an orange, all that was present, which was kind of cool. So then it kind of backed up what I said when I said, oh, yeah, there's other colors in there. Well, they got to see that. So this is where the light reaction is going to take place because we know chlorophyll traps light. So we're going to come over here and draw a little cycle like this and just kind of leave it for now, okay? All right, so once everybody's good to go, we'll start with the first step. We're gonna combine it to 4 steps and make it pretty compact. So what starts the whole process? What do you have to have? Sunlight. Other than chlorophyll, you've got to have some sunlight. So that's what we're going to put up here. Or we have to have light. We have to have photons of light. And we're going to put a number 1 right there, because what we'll do is we'll write the steps down below, or we can write them to the side, wherever you have the most room.
Teacher and staff, please release the following JV and varsity baseball players and managers at this time. Thank you.
At the time of day, I guess. Okay, we good? Well, they have lights, though. I don't know. I guess they all play on the same field, don't they? They have to play one game and then another game on the same field. Baseball takes forever. Okay, number 1 is chlorophyll traps light. So this is considered the light reaction. So there's 2 parts to photosynthesis. There's the light reaction that's light dependent, then there's the dark reaction that's the light independent. It doesn't mean it happens just at night. It just means it doesn't require light. So the very first one is chlorophyll. It's right in red. Maybe.
Or not at all.
Or not at all. That's right. It does not like me. Chlorophyll traps lights. Okay. All right. When it does, it actually excites the electrons. That's why there's little balls that are around here. And so when we acted this out in second period, we have chlorophyll traps light, and it gets the electrons inside of it so excited that it kind of moves them around. So that's the very first step. Second step is, what do we need to come into this chloroplast in order to make sugar? What are the 2 other reactants? All right, right. Carbon dioxide and water. Well, carbon dioxide we're going to find is going to come over here, but we do need some water. So take your blue, and you can draw like the little Mickey Mouse water, or you can draw a water droplet. It doesn't matter. Do you remember how many waters come into the process? 6. Right. So 6 H2O are going to enter the chloroplast. So that's number 2. Okay. So 6 waters enter the thylakoid. And when they do you remember when we did the carbon cycle outside, what happens to that water when it comes in? Light does something to it. It does. So it's called photolysis. So it's split by light. So enters, let's see, enter the thylakoid and split. Light. Photolysis. Okay, so when you see this word, photo, what does that mean? Light. And lice, if you lice anything, you destroy it. So all we're doing is splitting the water. So when the water is split, it releases the hydrogens actually to go make something important for the dark reaction, but it releases oxygen gas as a waste. So that's what we're going to put. It's split by releasing O2. Now, do you remember how many O2 are going to be released? Yeah. Releasing oxygen. And I know I'm kind of oversimplifying it, but this is really, if you understand this, you'll be good to go. So let's show that releasing the oxygen. So 6O2, come out. Well, we've made oxygen, we've used some water, we've used some light. What's left? Okay, so that starts part 3, the dark reaction, or step 3. So the dark reaction is going to take our CO2. Here's our third. Okay, and it goes into what we call the Calvin cycle. That's just another word for the fellow that discovered it. The Calvin Benson cycle. So we good? Can I move it down?
No.
Okay. CO2 goes into the cycle. Okay. So this is the dark. 6 CO2 fuse. Now, how did this CO2 get in there in the first place? How did y'all? What are the nostrils? The stoma, right? So, can the plants in that big tree out there without any leaves get any CO2? Can't. So, 6 CO2 diffuse into the stroma where the dark reaction takes place. Okay, so that's this. All right. And it's going to produce 1 glucose. So, okay, so my question, I want you to look at the diagram after you have that last step, and I want you to tell me where the atoms are coming from to make the glucose. Because I feel like there's enough of you that are left-brained enough to where you like to, like you're going to be the accountants of the world, or you like, you know, you need to make sure everything adds up. Because if you don't, it's like, okay, I can just believe her, which is fine, but I like to be able to trace it because that's how I understand things. So where do you think it happens? So tell me where the carbon comes from for the glucose. From the carbon dioxide, right? There's 6 of the C's and there's 6 C's. Okay, where does the oxygen come from? Well, most of that was released as gas. Okay, where else? We need 6 of them. From the carbon dioxide. But we have leftover, right? So part of the story is that some of this actually is released too. Where in the world did the hydrogens come from? The water. Okay, so this is the part that I'm giving you as honors. Yeah, so you see, that's why, that's as an honor student, you need to know this. Let's just put the hydrogens. So how many hydrogens does this glucose need? 12. And we get 12 from water, right? Okay, so I'm just going to do this. And it comes from the light reaction when water was split. Now, in second, that we talked about, well, hydrogen don't move on their own. You have to have somebody to kind of move them over there for you. And that's called a hydrogen acceptor molecule. So they will tell you, oh, it's NADPH, and that's great. But for now, as long as you know that, then you're in good shape. So if I wanted to monitor the rate of photosynthesis, how fast a plant was making glucose, what would be the easiest thing for me to count or to observe with my eyes? What product? What are the only 2 products you've got?
Water.
No, products.
Oh,
Oxygen is a product.
oxygen and glucose.
Can you count glucose being made by a plant?
You can.
Or could you count oxygen bubbles? And that's what we do. So this little lab activity that's actually in your packet, that's what it does. We're going to jump to it. It's pretty neat. But also, there's a question, and that's why it talks about oxygen bubbles. So that's the easiest way for us to monitor the rate of photosynthesis is to count the amount of oxygen that's made. Okay, so now questions about any of this business. Okay. All right, so y'all grab your computer